Hello students, welcome to today's video. In the previous video, that is the first video of this chapter, human reproduction, we had learned about the basics of the process of reproduction in humans. We have learned about the male as well as the female reproductive system. We had learned the primary sex organs, gonads, as well as the accessory sex organs in both male and female. We had learnt the ultrastructure of the seminiferous tubule, and we had learnt the location of the spermatogonia or the male germ cells. Similarly, we also had learnt about the female reproductive system, in which we had learnt about the structure of the female gonad or the ovaries, and in that we also had learnt the ultrastructure of the ovary, where we learned that it is the cortex of the ovary that you have the ovarian stroma, and that is a place where the oocyte is going to develop. Now we have learned what follicles are. Again, today we're going to learn in detail about all these things. And at the end, we have learned about the mammary gland and its importance in the process of the reproduction and also in the pregnancy, birth and the child care. Today, let us continue from there and understand the process of gametogenesis both in case of male as well as female. Now remember, the process of formation of gametes by the primary sex organs or gonads both in case of male as well as female is referred to as gametogenesis. The process of gametogenesis in male is referred to as spermatogenesis. Now what is this? The process of formation of sperms, which are the male gametes, from the immature male germ cell or spermatogonia is referred to as the spermatogenesis. Now, I hope you remember the structure of the seminiferous tubule, the ultrastructure, and if you remember that the inner lining of the seminiferous tubule contains the spermatogonia. As you can see here, these are the spermatogonia. Now, these actually develop into the sperms or the gametes, male gametes. How? Let us have a look. So the process begins at the puberty. I hope you all are aware of the term puberty, age of adolescence. You are learning it from class 8 and you should have a clear idea about it. Now remember, in the beginning the spermatogonia is going to be diploid, right? 2n. 2n or diploid means they are going to have 23 pairs of chromosome. Remember, 23 pairs of chromosome. Now, to increase in number, this spermatogonia undergoes mitotic cell division. Now, if you remember, mitotic and meiotic are the two types of cell division. Mitotic cell division is a cell division where there is increase in the number of cells without any reduction in the number of chromosome. That means the parental cell as well as the daughter cells will have the same equal number of chromosomes. If the cell was diploid, if the parental cell was diploid, then the daughter cell also will be diploid, and if the parental cell were haploid, the daughter cell also would be haploid. So there is no reduction in the number of chromosomes here. So the spermatogonia undergoes mitotic division to simply multiply or increase in the number. Remember, some of the spermatogonia, they specialize and function as primary spermatocytes. The primary spermatocytes undergoes meiosis. Remember now that the primary spermatocytes are also diploid. They are also having a 2n condition, 23 pairs of chromosome, and after they undergo meiosis. Remember, meiosis occurs in meiosis 1 and meiosis 2. It is reductional cell division. That means there is reduction in the number of chromosome in the meiosis 1. So during the first meiotic division of the primary spermatocyte, it gives rise to two equal haploid cells, and these equal haploid cells are going to contain only half of the number of chromosome compared to the original primary spermatocyte. That is, now they contain only 23 chromosomes and not 23 pairs of chromosome. Now after this, the primary spermatocyte, now referred to as the secondary spermatocyte, having a haploid condition, undergoes their meiosis 2 division or the second meiotic division, and here they will form 4 equal spermatids, and remember they are haploid. So meiosis II, there is no reduction in number of chromosomes, there is simply mitosis, and that's why they'll form 4 equal spermatids, and each of these are going to be again haploid in nature. After the spermatids are formed, they're transformed into spermatozoa, and the process is called spermiogenesis. Right, we'll talk about this process in a minute. After spermiogenesis, the heads of the spermatozoa, that is the structure formed after spermiogenesis from spermatids, they get embedded in the Sertoli cells. Now the Sertoli cells will help in maturation of this spermatozoa, and they'll develop into the sperm. Now upon maturation, the sperms are released from the seminiferous tubule by a process called spermiation. By the process of spermiation, the sperms are finally released after maturation from the seminiferous tubules, and they are going to be now transported via the accessory ducts to the outside of the male body. Now on this side you can see the picture that is depicting the sectional view of the seminiferous tubules. We had said that the seminiferous tubules, this is the wall of the seminiferous tubule. So near the wall or the inner lining that we are saying, you can see the spermatogonium here. Now the spermatogonium, as we are saying, undergoes mitotic division from the primary spermatocytes, that undergoes the meiotic I division giving rise to secondary spermatocytes, and that undergoes the meiosis II to give rise to the spermatids, and finally by the process of spermiogenesis it gives rise to the spermatozoa. Now the process of transformation of spermatids to spermatozoa is important, as during this process, as you can see here, the sperm's actual structure is going to be developed. Now you can see here, this is the entire process of spermiogenesis and how the transformation is occurring from the spermatid to a typical sperm. Now you have learnt in the junior classes a basic structure that the sperm is consisting of a head, middle piece and tail. Now you have to understand that there is going to be presence of a specialized enzyme called acrosome, and this acrosome is going to be developed from the Golgi apparatus. So when the spermiogenesis begins, the spermatids Golgi apparatus, they start developing or secreting the acrosome, and they form the acrosomal vesicle, and slowly the flagellum is going to form. The mitochondria are going to organize into the middle piece area, and the head takes its shape with the haploid chromosome set or nucleus in the center, with the tip with the acrosome. Remember, the acrosome is going to be very important, and we'll talk about its importance in a later part. So here, as you can see, a regular cell is going to be transformed into the sperm with a characteristic head, middle piece and the tail. Now let's understand what are the hormones that control the process of spermatogenesis. There is the involvement of hypothalamus. So during the puberty, the hypothalamus releases the GnRH. GnRH stands for gonadotropin releasing hormone. Again I repeat, gonadotropin releasing hormone. And this GnRH, it acts on the anterior pituitary, and when the anterior pituitary is stimulated by the GnRH, they release the gonadotropins. Right. So what are the gonadotropins? There are two. One is the LH or luteinizing hormone, and the second one is called the FSH or the follicular stimulating hormone. Now remember, the LH acts on the Leydig cells. Leydig cells are also known as the interstitial cells, and they will produce androgen, and this androgen is going to stimulate the process of spermatogenesis. On the other hand, the FSH is going to act on the Sertoli cells that are present on the inner side of the seminiferous tubule, and they produce certain factors that is going to help in the process of spermiogenesis. So you have to understand that there are two hormones. One is called LH and the other one is called FSH. Both of these are gonadotropins released from the anterior pituitary when it is stimulated by the hypothalamus during puberty, and the LH is going to help in the process of spermatogenesis, whereas the FSH is going to help in the process of spermiogenesis. Now let us have a look in this particular image. As you can see here, the hypothalamus releases GnRH that acts on the anterior pituitary to release FSH and LH. The LH is going to act on the interstitial cell or Leydig cells, and they will help in the release of the androgen of the testosterone, whereas the FSH is going to act on the Sertoli cells to produce certain factors that is going to help in the process of spermiogenesis. The Sertoli cells also going to release the inhibin. Now why it is important? Inhibin is going to be sending negative feedback to the anterior pituitary. Now why it is important? This inhibin is going to help in regulating the amount of FSH that is going to be secreted. The testosterone also negatively provides feedback to the hypothalamus and pituitary, thereby inhibiting further release of GnRH, that is gonadotropin releasing hormone, FSH and LH. So here there is going to be the inhibin that is going to have a negative feedback on the secretions of the FSH and LH. Now understand the structure of the sperm. Now first of all, you have to understand that the entire structure of the sperm is a unicellular structure, and as it is a unicellular structure, the entire surface of the sperm is covered by the plasma membrane. It is consisting of three major components: head, middle piece and the tail. Now the head is going to contain the haploid nucleus, that is, as you can see here, this area is the area that is going to contain the haploid set of chromosome, that is the 23 chromosomes, and this is going to be anteriorly covered by the acrosome. As you are saying, this is the enzyme that is going to play an important role in the process of fertilization. So acrosome is present at the anterior position, and it is essential for the process of fertilization to occur. Now the next structure that actually is present between the head and the middle piece is the neck. This connects these two structures. Now the middle piece is extremely important as they contain the mitochondria, as we discussed, and the mitochondria is going to help in providing the energy, right? They produce the ATP, and this is going to help in the sperm to swim across and reach this target site, that is the place of fertilization. Remember, the mitochondria is going to use fructose present in the seminal plasma to derive energy in the form of adenosine triphosphates. Now remember, the tail is going to be the last portion, and it is going to help the sperm in the motility. Now a single ejaculation during coitus, around 200 to 300 millions of sperms are released, and for a normal fertility of these 200 to 300 million sperms, at least 60% must have the normal shape and size, and at least 40% should or must show vigorous motility. That means they should have the ability to move 40%, and the remaining 60% should have the normal shape and size. Now let us understand the pathway of the sperm transport. The matured sperm detaches from the epithelium of the seminiferous tubule, and it is going to enter the lumen or the cavity of the seminiferous tubule. Now from the seminiferous tubule, it is going to move to the retrotestis, and then to the vasa efferentia, then to epididymis, and then to the vas deferens. So till this point, the sperm was traveling alone, and now the secretions from these accessory glands, that is the seminal vesicle, prostate gland and bulbourethral gland, they produce the seminal plasma, and this seminal plasma mixes with the sperm and produces the semen, and the semen then moves to the urethra and it is going to be released outside the body. So as you can see here, seminiferous tubule, reticulosis, vasa efferentia, epididymis, vas deferens, and from there it is going to receive the seminal plasma through the ejaculated duct, and then it is going to be called the semen, and then it is going to move to the urethra, and from here it is going to go to the outside of the body through the urethral meatus. Now let us summarize the entire process of the gametogenesis in male or the spermatogenesis. So at the beginning we had this spermatogonium that is diploid in nature, that underwent the mitotic cell division to increase the number. Some of these, they acted as the primary spermatocyte underwent the meiosis 1 to give us the haploid, right, 1n or haploid spermatocytes, secondary spermatocytes. These secondary spermatocytes underwent the meiosis 2 to give us the spermatids, again which are haploid. Now by the process of permeogenesis, the spermatids got transformed into the spermatozoa that got embedded into the Sertoli cells to give the mature sperm. As you can see here, this is a cross section of the seminiferous tubule under a microscope, and you can see different cells in here. For example, this is a layer, right? This is the layer of the This is the layer of the seminiferous tubule, and here you have the spermatogonia. Here all of these are the spermatogonia. Here you can see the primary spermatocytes, right? And you can see the Sertoli cells in here. These are the Sertoli cells, right? And also you can see these are the interstitial tissue or the Leydig cells. And here also you can see there are different type of Sertoli cells. And here you can see this particular area, you can see the early spermatids, and the process continues. You can see some of the different structures that we're discussing when in this picture, and this is taken from a microscope. This is how it looks under the microscope. So that completes our understanding on the gametogenesis in male or the spermatogenesis. We will continue the discussion on the remaining concepts in the next video. Thank you all for joining today in this video.